Now I'm next very pleased to introduce Catherine Scart, who's the Chief Medical Officer of SOM Biotech. Thank you, Catherine.
Thank you. So first, I want to thank the committee of EHDN for inviting me to present our project and SOM Biotech as a new actor in the field. So to briefly introduce SOM Biotech, we use artificial intelligence to select drugs that can be repositioned on new indication in rare disease with a new mechanism of action and allowing faster development to propose to patients a new therapeutic option. For Huntington disease, we have first taken tetrabenazine as reference drug to search candidates with similar mechanism of action. And with artificial intelligence, we have selected bevontorol as the most VMAT2 inhibitor, most potent VMAT2 inhibitor. Bevontorol was initially known as a beta blocker used for years as antihypertensive drug with no psychiatric problem. So it could be safer than tetrabenazine. After preclinical stage, where we have assessed the VMAT2 inhibitory activity, the brain penetration, and the effect on CNS in animal models, we could test it directly in clinical proof of concept, which has confirmed that the drug that we call bevontorol, we call it SOM 3355 with this new development, and we had the confirmation that it reduced chorea in patients with Huntington disease. So now we are launching a pivotal phase 2 study, and I will present you this study. So in terms of mechanism of action, bevontorol is acting as a potent VMAT2 inhibitor, and it's as a dopamine depleter, so it alleviates the hyperkinetic symptoms such as chorea. In vitro assays, we have shown that SOM 3355 inhibits VMAT2 with the same potency than tetrabenazine, but it interacts differently on the VMAT2 receptors. So we can differentiate our drug to tetrabenazine, and also we inhibit the VMAT1 while tetrabenazine doesn't. So it could explain the preservation of mental health. Bevontorol is a well-known drug. It was a beta blocker developed in the 80s, and it is currently commercialized in Japan and in Asia for hypertension. And this drug, after more than 30 years of use in hypertension, has a very good safety profile with no concern regarding the CNS side effect, and particularly no signal in terms of depression and suicidality, which could be interesting. We have been granted orphan drug designation for the treatment of Huntington disease with this drug by the FDA last year. SOM 3355 was first tested in a proof of concept study. It was a small study conducted in Spain in 4 sites, and 32 patients were with mild to moderate choreic symptoms were included in a double-blind randomized crossover study comparing SOM 3355 with placebo in a crossover design. So the primary endpoint was the improvement of choreic symptom measured by the total motor score, and we considered at least 2 points of improvement as significant. And with the active treatment drug compared to the placebo. We had also several other endpoints. So SOM 3355 have shown, we have confirmed with this study the effect on chorea, and we have shown that in this study, quite 60% of patients had an improvement of at least 2 points on the TMC score, and some others had more. On the 200 milligram BCD dose has significantly improved the choreic symptom compared to the placebo. So this was a confirmation that the drug is effectively reducing choreic symptom. In terms, we have also shown that the good safety profile was confirmed in this new population of Huntington disease patient with good safety profile, with the low rate of adverse event, and all the adverse events were manageable and expected with the known mechanism of action of the drug as beta blocker, but we had no case of depression or suicidality. And in terms of vital signs, we had a slight reduction of the heart rate and the systolic blood pressure, but still in normal range and well tolerated. No patient had to interrupt the treatment. In this study, we have measured the prolactin plasma level, which was slightly increased, and this is reflecting the effect of the drug on dopamine pathway. With PKPD modeling, we have predicted that a higher dose, such 300 milligram BCD, may have a stronger effect and with the same, at least the same magnitude of tetrabenazine. So we have discussed with the EMA and the FDA the design of this study, and we are now launching this pivotal study to assess the real treatment effect. So it's a phase 2 randomized double-blind placebo-controlled study in parallel arms, assessing 2 doses of SOM 2355, 300 milligram BCD and 200 milligram BCD, compared in parallel with placebo. The study duration is 12 weeks, and starting with 3 weeks with an up-titration during 3 weeks, and then a maintenance dose for 8 weeks. The treatment will be given orally twice daily, with 1 capsule in the morning and 1 capsule in the evening. The objective of the study is to assess the efficacy of the 2 doses compared to placebo in reducing chorea on the total maximal chorea score, which is the endpoint accepted by the authorities. And of course, we have to check the safety and the tolerability of the 2 doses compared to the placebo, with a particular attention on depression and suicidality, and also on the cardiovascular parameters. There is also other key endpoint, and we have also a sub-study that will be conducted on a subset of patients to determine the PK profile of the 2 doses and perform some PKPD assessment with cardiovascular parameters. In terms of selection of the population, we include the patient. We will include adult patients with a diagnosis of Huntington disease, suffering from chorea, with a total maximal chorea score of at least 2 points. And those patients should be mildly to moderately impaired, with a TFC score of at least 7, and they should be able to work independently, and they should be also able to give their own consent to participate in the study. We will exclude juvenile form, patient with rigid akinesia, and the patient already treated with antichoric medication that may interfere with the study drug. We exclude also patients with severe depression and active suicidality, and also on patient with history of such event. We exclude patients with known hypotension or bradycardia, and also any active clinically significant illness. Sorry, I missed. So during the study, for the concomitant medication, all the other antichoric treatments will be prohibited. Tetrabenazine and derivate, of course, because they have the same mechanism of action, and the neuroleptic given to treat chorea because they are also dopamine depleters and the other treatment. And I must say also that we authorize all the antidepressant to treat the treatment if the drug is given at stable dose during the whole study. And for neuroleptic that are needed for aggressive behavior, they will be accepted on a case-by-case basis. So the endpoint, the primary endpoint is the change in total maximal score between the baseline and the maintenance dose. And this study will be conducted in 7 countries in Europe. You have the list here with 22 sites. And the study is now starting. We have all the approval in all the countries. We had the first patient enrolled in August in Spain, and all sites are currently initiated, and we consider that all the sites will be initiated by mid-October. The recruitment is expected to be completed by end of March, to have a study completion by end of June, to be able to present you the first top-line result next year in September. For the patient recruitment, we are working in a close collaboration with the EHDN and the CHDI, and also with the patient association, because we consider that it's very important to involve also the patient in this project for the awareness. And we will have patient engagement officer who will liaise with the patient association, and the site manager who will liaise with the site to facilitate the patient recruitment and solve all the logistic issue. And to end this session, I want to announce you that we are also planning an open label extension study for the patient having completed this study, and they will be offered to beneficiate of the active drug for a year with a visit every 3 months. Thank you for your attention.
We've got time very quickly for one question for Catherine. Do we have any questions before our next speaker? Any questions? Oh, Michael.
Thank you so much.
There's a microphone.
Okay. Thank you so much for that presentation. I'm just wondering about the selectivity compared to Austedo and valbenazine, which are other antichoric drugs, one approved, one close to approval.
Sorry.
Selectivity compared to tetrabenazine and valbenazine, which are
Austedo, deuterated tetrabenazine.
Yes, we are studying this, and we will have a further element on this.
Okay.
Yes, this is something we are studying.
Lovely. Thank you.
Thank you.